Yeah, hi everyone, and welcome for the first hybrid session. So as I said, it's the first one. We had a test run a few weeks ago. In case there are any technical problems or other things going on, it don't really work today, so apologies. It's the first time. So yeah, today we have Dimitrios Chatziplis, I'm sorry, as guest speaker. He is a professor for animal genetics and breeding from the Hellenic University in Greece, and he's currently on a sabbatical in Roskilde for 1 year or for how long?
Until September now.
Oh, okay, until September. Good. He's a quantitative geneticist who has worked in all farm species, including fish, and also in plants. And the lab he is currently leading is involved in several national and international projects on fish breeding. Yeah, if you're ready.
Yeah.
I'm just kidding. Thank you.
Hi, nice to see you all. I've been here many years ago as a PhD student, so I'm back for a few months left now for my sabbatical, working with GWAS and Enrique Sanchez on Epistasis, how to identify it and how we can utilize it in genetic evaluations. Where are you now? Let's see. For example, now we're using a infinitesimal model where we assume the genetic effects are a sum of infinite number of loci, and this model was used for many years now. This application into predictive theory in animal breeding. However, we tend to ignore that there is dominance and epistasis for reasons that explained in several reviews, mainly also by my ex-supervisor Bill Hill that passed away. God bless his beautiful soul and mind. And this model can predict, for example, it cannot predict likely limits to selection, like we have but only in simplified condition, like the Palmer effect, and also cannot predict changes in reproductive fitness that might be expected accompanying selection. Also, it's only additive. What about dominance and epistasis? We're ignoring it when we're using it. When I was born, that shows how old I am, and Enrique can teach me even more now. The supervisor, my supervisor, and one of the first directors in animal breed research organization at the time, he says that in order to overcome these deficiencies of the infinitesimal model, it's necessary to have a description of the phenotypic variance in terms of individual loci affecting a trait. And he said that the description doesn't have to be full, just to be, it doesn't have to be complete, but at least have some, explain some of the major effects. And also says, this description have to demonstrate a small number of genes have a major effect on traits, and also map those genes, and also, if possible, to identify the mode of action given the existence of the genes. For the first two, the last few years, quite a few years now, we're managing well. We're building on that. The third one, we don't have so much work. It's only the last few years that we're working on it. But let's start with definition, what we call epistasis and what would be their effects on animal breeding. Is the third component, as we in the row where we put it in the phenotypic variance formula. And one definition that I found really easy and very convenient is by Churchill, is that epistasis is defined by the genetic interaction of two or more loci to produce a phenotypic output that is not predicted by the additive combination. So that's not predictive, it cannot be predicted by the additive combination. And in the literature, you can find many types of epistasis. I had about 6, but Enrique said to reduce it just to give it just 2. And the duplicate would be dominant or recessive, polymeric gene interaction, also recessive, etc. There are about 6 the main. But we'll see, concentrate in the duplicate, with example of having a simulation is a duplicate epistasis. Another more quantitative genetics or more statistics approach of the of a definition is by another guru of genetics, Mackay. And he said in a review paper in Nature Genetics that epistasis is a modification of additive and non-dominance effect due to loci interaction. So it's actually considered like it's modifying the additive effect. And he said that it can be visualized by plotting the phenotypes of the 9 different genotypes. And that the epistatic interactions can be fall in 2 categories. One that is a change of magnitude in which the phenotype of locus either enhanced or suppressed by the genotypes of the other locus, or a change of direction of effects. And he saw it in his review, the graphs like that. What in the first one we have additive, only additive, but no epistasis. And I put a question mark like that because you can have epistasis, similar graphs with epistasis. And what we'll see later, what Mackay called pervasive epistasis. The second one is the enhanced effect. I would say the change in magnitude, where, for example, the additive effect of Y locus is much greater on the homozygote for the 2 allele genetic background than the homozygote for the X1 allele genetic background. And then we have the additive effects of locus. The third one, which is the duplicate epistasis, the additive effect of locus X are opposite in the Y. Sorry about that. Continue now. Okay, the question is why we detect with GWAS, we detect very few times major effects. Mackay gives one possible explanation is that rare alleles have large effects. They're relatively new mutations compared with the common with other alleles, and that epistatic modifiers that ameliorate the effect have not been yet occurred in the population. The same idea could hold, for example, in the case that we select our training population. For example, when we select our training population, sometimes we might not select the epistatic modifiers, and we have the appearance of the additive effect, but they're not cancelled by any epistatic effect. Or there are very few, or none at all. Very good example is when we use selective genotyping to genotype our training population. So you use parameters, trait parameters for the selection of our samples, then this one might appear. Now, how epistasis can affect the selection? It can create biases in the estimates, the genomic or genetic parameters. It can affect the accuracy of the genomic estimated breeding values, and as Mackay said, that you can have in the presence of epistasis, you can have poor predictions, genomic predictions. It can affect the accuracy of genomic selection, and therefore have an impact on the long-term genetic gain of genomic selection. And it was shown that would be the case in a simulation example that they done some guys from Netherlands, Wientjes. I don't know if I pronounce his name well. Anyways, with Peter Wisma. I know him from the old times. Let's see examples where I noticed from previous studies that are done that epistasis might have an effect in detecting major effects. In the study 1, we use a complex segregation analysis, no markers, and we had indication of a major gene segregating in 2 different populations of sheep of Chios milk breed for milk for milk yield, one in population in Cyprus, one in Greece, and we estimated the major effect, the major gene effect, between 10.5 to 11% of the additive genetic variance. Then using this segregation analysis from the posterior distribution, because a Bayesian analysis, we select the heterozygote sires and we took the genotype their daughters for candidate genes. And we ran an association analysis using mixed model, having a polygenic component also in there, and that revealed an SNP on exon 10 on acetylcoenzyme and acetyltransferase 2 that had an effect very close to the one that we estimated with a more rough segregation analysis. The estimated effect was about 10 to 11% of genetic variance. Then the same colleagues have done another study a few years later, and in another population, in a commercial flock. The first one was in two nucleuses population, state nucleus populations. The third one was on a flock, a commercial flock. And they found that, in effect, that on the same SNP on exon 10, on the ACA2 gene, but the effect that the estimate was much smaller, was between 0.62 to 2.62%. And also, the seen differential allelic imbalance expression. However, in the same population of Orford, the initial analysis, I had genotype also some SSR, some microsatellites, in order to estimate inbreeding. But some of the microsatellites I chose that were near QTLs from other breeds. And when I ran a mixed genetic model, an association model again, I found that some microsatellites that were near QTL, they have an effect as well on the trait as well as the SNP. But they were taking variation out of the SNP effect. Then I've done master students. I gave him a project, master thesis. We sampled some 2 farms with the same breed, and we genotyped the exon 10. We sequenced the exon 10. We found identified the SNP there, plus the microsatellite markers some near QTL in other breeds, and we fitted the interaction between them. And there was significant interaction effect with two of the SNP with 2 microsatellites, both of them near QTLs. The one was for QTL for milk on chromosome 19, and the other one, chromosome 17, near QTL for seasonality. I chose this QTL for seasonality because Hughes breed is the most seasonal breed in the world. So both those 2 microsatellites, they saw the express interaction with the SNP in acylotransferase gene. Oh, sorry. And later on, a year ago, Valentina, in a genome-wide association analysis, had a significant effect on milk yield on chromosome 19 near an SNP near the same microsatellite. So that makes me wonder if the whole missing effect, the large effect we had initially, was due to epistasis. Another example is from parasite count in fish this time, in SIPAS, in parasites of gills. And we done a genome-wide association analysis, and here we can see, for example, only 2 SNPs, one at the beginning, one at the end of chromosome 8, that you think, oh, most probably is false positives. This is your first thought. If we estimate the effect of these 2 SNPs, there are 2% of the phenotypic variance each, very low effect. However, if you fit the interaction between them and also with some other SNPs, you have significant effects on the parasite count. Then let's see another example from fish, from Cibrim this time. A genome-wide association analysis on slaughter age, for body weight or slaughter age. That picture will tell you that is most probably is polygenic, the trait. But the question is this is because polygenic by small additive effects, or there is epistasis exists? We select based on the 1%, based on the threshold of 1% with no bound correction, the most significant associated SNPs. This is the selected it was 1,180 SNPs. The total population, notice, based on selective genotyping. We estimate the genetic parameters from the whole breeding program, about 20 years of data, and the heritability was 0.28. Using the GRM from the selected SNPs, about 1,500 records, we estimate the genomic heritability of 0.29. When you use the full SNP, only because there were 1,500 fit with, because it was a program for estimating G by E interactions, also fit by E interactions. When you use the control fit, we have an estimate of heritability of 0.26. However, when you use both fits, we have a strange heritability estimates. We're still investigating why there is so strong G by E interactions, and if they are not only genotype by environment interaction, but also epistatic effects that they change according to them. So it's something we're working on. However, with the heritability the second heritability, the 0.29, it seems that we can capture the additive genetic variation that exists in the whole population for 20 years in a window of time. So sampling 1,100 SNPs, selected, using the GRM, and get the same estimate of the heritability. But is that a right amount of genetic variation we capture? Is it the correct genetic variation? It seems that that's the case. Now let's see other questions to be answered from additive genetic variation model analysis. The one we asked before, can we estimate the correct genetic variation? We can with the infinitesimal model because of limitations stated many years ago, with exceptions of utilization of dominance using BLAB, done quite a few years ago. In genome-wide association analysis, it can be used to capture the additive genetic variation or confounded variation, for example, like pervasive epistasis, you can catch it. And also, you can use genomic relation matrix or SNP effect to utilize them in genomic selection. Can we fit interactions? Too many. There are too many interactions. Only if you get the second order interactions for 1,000 SNPs, it's about 500,000 interactions. Can we identify which ones are important from these interactions? We can use complex statistics, either in genome-wide association or in genomic selection methods, in which you can fit the interaction terms. There is a nice review by Luis Varona in 2018. However, you need high processing computer power, very intensive, even for second-order interaction. You go to third-order interactions, it's even more difficult. Maybe a good analysis for complex diseases in humans, where you have more money for research, and might be easier. For animal breeders, it's a bit difficult. Can we use empirical approaches? Can we filter the important interaction in order to include them in a more representative genetic model for our genetic evaluation? We don't want a perfect explanation of the variation, just a better explanation of the variation, actually. Can we answer the previous questions? We use a simulation program, some of the prediction process, the research is still going on. We use Fortran. The exclamation mark is because they call me dinosaur, me and Ricardo, still programming in Fortran. We create a base population where we use it for burning period for 500 generation, and then we sample a generation 0 from there. We create the gametes, we're mating animals of generation 0, constructing the chromosomes of generation 1, then the same to the generation 1 animals, creation of gamete, mating, construction of chromosomes, and generation 2. In the generation 2, we assign the phenotypes also according to the genotypes. We use 60,000 SNPs, simulated 60,000 SNPs. They're equally distributed in 24 chromosomes with an average distance of 0.05 centimorgans between them. The tape is controlled by 4 additive QTL, 2 pairs of epistatic QTL. The epistatic effects are outside of the heritability, the narrow sense heritability. They're not included in the additive genetic variance. And also we have a polygenic component, whereas the rest of the variance, if you subtract the additive variance, you can subtract the variance of the QTL. But the polygenic component is simulated by 10B allelic genes and is unlinked and unrelated to the 24 chromosomes. They cannot be captured by the SNPs, by the SNPs. And the question is why you did it this way. We can discuss it later. The additive QTL variance is expressed as a percentage of the additive genetic variance, a percentage of the heritability. The frequency of the QTL alleles, there are 4 additive B allelic QTL on 2 chromosomes, one on the beginning, one on the end, with equal frequency, and 2 pairs of biallelic QTL of equal frequency for epistasis. Their effect is also, as I said, dependent on the heritability of the trait, the narrow sense heritability. The frequency of the 10 dependent genes are also biallelic. They are unlinked with each other and with the QTL, and they are independently inherited. They are biallelic with equal frequency and not included or mapped in the genetic regions of the SNPs, the genomic regions of the SNPs. For example, here we see on chromosome 12 and 24 is the additive QTL, one at the beginning, one on the end. And on chromosome 2 and 8 are the interactive QTL, the epistatic QTL, one at the beginning, one on the end. And also we include interactions only between E and G and F and H. We don't have between E and H and F and G because we saw that we have tertiary effects and additive effects on that, so we want to exclude it through this case. And also they are not within chromosome interactions. So consider a very plain model. Only 8 interactions are actually considered. We run 20 replicates with quite large heritability, 0.4. The 4 additive QTL, that's the genotypic value is coming from Wheeler for Falconer. 70% of this heritability is due to the QTL, so it's about 28% of the phenotypic variance. Actually, 7% each QTL. There were 4 additive QTLs, 7% each QTL of the phenotypic variance. And also we have 2 epistatic QTLs. Their effects are not included in the additive genetic variance, and we'll see later on we use different scenarios of different levels of epistasis. The polygenic effect, as we said, is 10 genes unlinked and independently inherited of the QTL, and they're mapped outside the area where the SNPs are mapping. 3 different scenarios of epistasis, one with no epistasis, only the additive QTLs are affecting the trait. Second one, medium epistatic effects. Each epistatic effect is a quarter of the additive QTL effect, so 1/4 of the 7% of the phenotypic variance, for example. And we have high epistatic effect. Each epistatic effect is equal to the additive QTL effect. We see an example of the epistatic effect. I put it also in terms of alleles. When we have interaction between allele E1 and G1, it's actually will be, according to the scenario, 0, a quarter, or the additive effect of A1 and A1. And if you see it in terms of genotype, it's 4 times that effect in terms of genotypic effect. And the same equal effect also in the other pair. Here we can see the heterozygotes has the opposite effect of the homozygotes, and the homozygote for the second allele has 0 effect. We follow this model in order to create, for example, the third case of Mackay et al., where we have the exact change of direction in the effect. For the simulation results were quality controlled by Plink, and we've done genome-wide association using GEMMA. We used 992 animals from 124 families, full SIP families. The family size was 8. Now the results, the power to detect QTL in terms when we don't have any epistasis is about 75% to detect at least in one QTL. To detect both QTLs is 15%, quite low, both QTLs. Also, in type 1, as much as the type 1 error, we detect an epistatic QTL when it doesn't exist, so it was just by chance. The estimated effect is not so good. Usually, it's putting all the variation in one chromosome where the 2 QTLs are lying. So, in total, we're overestimating the QTL effects. And also, you cannot distinguish between the 2 QTLs within the same chromosome. When in the existence of medium epistasis, power is drop dead. As you can see, it's almost 0, only 5% to detect at least one QTL, and that will be the additive one. When we increase the epistatic effect, we have very good power to detect at least one QTL, but is the epistatic, that will be the epistatic, 94%. And the power of the additive to detect additive QTL is quite low, it's 22%. Again, the estimated effects are not so good. They are in many cases, in one chromosome, they're very much underestimated, and the additive QTL, they're all underestimated. In the case of epistatic, is putting all the variance due to the QTL on the QTL that exists in chromosome 8 in that case. These are some examples of the Manhattan plots that we see. For example, I put 6 replicates out of the 20 where there are no epistasis. In that case, in the first row, you can see where both QTLs are detected on chromosome 12 and 24. In the bottom row, there are one case where it's detected only on chromosome 12 and only on chromosome 24. And the last one is where we don't have any detected QTLs. In the case of when we have the epistatic effect is a quarter of one additive QTL effect, we didn't have any power, so the only one that detected one QTL, it was on chromosome 12, only one replicate. In the case of high epistatic, strong epistatic effects, we have a picture here where we have detected one QTL epistatic, will be the epistatic one on chromosome 2 on chromosome 2 in the first one, the second one on chromosome 8 and the third chromosome 8. The bottom row, you can see detection of 2 QTLs, both epistatic on chromosome 2 and 12. Here one epistatic and one additive, and here 2 additive. That's a general picture of the Manhattan plots. In the absence of epistasis, we have medium power to detect at least one QTL, about 75%, and low power to detect multiple QTLs. Genome-wide association analysis cannot distinguish different QTL on the same chromosome. The estimated effects are not accurate. Usually on chromosome in which the QTL map is absorbing almost all of the variation controlled by all the QTL. However, this one might be effect of the power of the design to few animals, for example. In the presence of medium epistatic QTL effects, extremely low power to detect any QTL, 5%. If a QTL is detected, it will be the additive. The QTL also absorbs all the variation from the other QTL in the estimated effect. In the presence of high epistatic QTL effects, we have very high power to detect at least one epistatic QTL, 94%. However, low power to detect epistatic QTL in both chromosomes, 11%. And low power to detect also additive QTL. GWAS QTL detection cannot distinguish different QTL, whether they are epistatic or additive. Everything is an additive model, so everything is detected as additive. The epistatic QTL, for example, are detected as additive. In the presence of high epistatic QTL effect, as in the presence of in the absence of epistasis, the estimated effects are not accurate. We saw both in the case of both additive and epistatic QTL. Usually, one of the chromosomes is getting all the variation. What will happen if we increase the power of GWAS? We double, for example, in this case, we double the sample size of the analysis. We use approximately 2,000 animals. Then in that case, the additive, in the case of the additive with no epistasis, we have increase in power, 85% to detect the least one QTL, 55% to detect both QTLs. And also, in the case of the presence of epistasis, we increase also the power. It was 0 in the previous time, now it's 83 to detect 5% in the previous time with half of the sample, 83% now to detect at least QTL, this one QTL, and 33% to detect both additive QTLs. And also, it's increased drastically the power to detect the epistatic QTL, 56% to detect at least one and 6% only to detect both. In the scenario of high epistatic effects, the power to detect the additive QTLs is going really down, 33% at least one QTL and none to detect both QTLs. However, the power to detect the epistatic QTLs is going very high, 100% to detect at least one, 50% to detect both. But In both cases, the estimated effects are not improving great. We saw, for example, when we increase the power by increasing the sample size, we have improved power to detect the additive QTL with large sample sizes. However, in the presence of strong epistatic QTL, power to detect the additive QTL is better, well before, but still very low. It's only 33%. Power to detect epistatic QTL with strong effect is increasing drastically when we increase the power of your study, increase your sample size. We have low accuracy of the estimated QTL effect, usually on the chromosome again is getting all the variation. However, high power genome-wide association is quite costly. If you have the money to genotype 2,000 animals, it's good, but usually that's not the case. In the presence of epistasis, in conclusion, power to detect QTL can be diminished. It went down to only 5%, for example, to detect one QTL when we don't have enough power, when we have sufficient sample size to secure good power. It seems that it's masking even the detection of additive QTL, the presence of epistasis. When there are strong epistatic effects present and medium sample size in genome-wide association analysis, the epistatic QTL are most likely to be detected. However, the QTL are detected as additive because it's an additive model anyway. And pervasive epistasis is consistent with additive variance, like Dr. Mackay says. So you're actually detecting this pervasive epistasis with an additive model. This is what happens. But the power to detect QTL, any QTL is minimized, any additive QTL is minimized when there's stock epistatic effects. And the estimated effects are not accurate in the presence of epistasis. Maybe because of the small sample size, but as we say, even when we increase the sample size, still the estimated effects are not accurate at all. Usually the one chromosome is taking all the variation. Epistasis seems to add noise in the genome-wide association analysis. It might not detect any QTL, even those with strong additive or epistatic effects, when sample size does not provide sufficient power. GWAS methods might not sometimes work for QTL detection in the presence of epistasis. So when we make inferences, I didn't find anything in the genome-wide association analysis, that might not be true. It's not always the case. It's not always say, oh, it's polygenic. Might not be the case. Might not be correct, that statement. The presence of epistasis might also have an effect on genomic evaluation methods. There are publications that show that. So better, one good thing to do is to investigate alternative methods or fine-tuning existing methods in order to improve our evaluation, genetic evaluations. Such methods could include selection of SNPs that significantly influence the trait, filtering significant interactions between SNPs, and utilize haplotype inverted or combined genotypes of these SNPs in our genetic evaluation models. So that's one option to do it. I would like to thank the Roche Institute for having me for my sabbatical and Professor George Banos. Also, I would like to thank Enrique for all his help and fruitful discussion, but most of it for his friendship, and also Ricardo for his fruitful discussion in programming in Fortran and genetics algorithm, and also for his friendships for about 30 years now, Ricardo. Okay. That's it. I cannot see the questions from here. I cannot see questions.
Yeah, I have my laptop. So I see them fully from here. Yeah, thank you again for this very interesting talk. Are there any questions online or in the room so far? Yeah.
I was just wondering, when we started off with the whole QTL mapping,
Yeah.
when we originally knew each other, then there was this thing that we used to do. If a QTL was marginal, we'd fit it as a fitted effect and then scan the genome, see if anything else popped up. Two parts. Why don't we do that? And how would it affect epistatic QTL? If you were able to fit the main effect, would you actually find others?
Yeah, it's for example, as well, you see, I want to do this. I've done it actually with microsatellite markers, fitting the one material you find and scanning the other one, and scanning also if there is interactions. That's one good way. But it will get too much computer power if you do it in GWAS because there are too many markers. So you need some way to filter them to avoid, for example, too many comparisons. It's a computational problem. If you go and do it the whole thing, your H matrix is getting absolutely massive. You know, you're needing a lot of power. If you are, for example, in medical research and you have huge cluster, you might be able to do it and you leave it to run, but for us, it will be difficult. Maybe for Rosalind, yes, but for us, less poor people, poor labs, that we don't have big clusters, it will be almost impossible. We can run it, for example, in association, but still it's quite big job. It's too many markers.
Yeah.
If you find a way to filter them, do a pre-selection first, and then fit of these ones, and from these ones select. There are ways of reducing the H matrix.
And another question for the impact on GEVB, would we tend to underestimate
From what,
the variance
yeah,
explained, or
from what I've seen, usually is overestimation. Usually, like, for example, when I'm suspicious of epistasis, I'm seeing an overestimation. But what I've seen now in the simulation, when you have multi-level interactions, when you have not only, for example, between tertiary effects and that one, is inflating too much your phenotypic variation. So it's decreasing your denominator is increasing, so it's increasing your ratios, your heritability estimate. So it has strange effects. And also, my suspicion is will depend also what kind of epistasis you will have. If you have change of magnitude, you have different. I will expect to have different effects on your estimate, either genomic or genetic heritability. You have a cross one, like we say the duplicate one, you have different ones. This is what I believe. I haven't checked it out. We didn't reach the stage of the effect on the genomic evaluations. That's the next step, actually, we'll do with Enrique.
and I guess it's also what you consider to be the wild type for the
Yeah.
yeah.
Yeah. But it's strange, for example, with selected SNPs, we tried in another scenario to estimate the genomic heritability, and we still got a very good estimate. So it seems that how you select your SNPs, it seems to have an effect on your estimates. So that's very, very early observations. The next step is we'll see it now with simulation and see, I will tell you, for example, the next few months, once we've done it, I can tell you exactly what and how. Because if you sit in simulation, you know what you simulated, so.
Can you, sorry, is this finished? Oh, I do, okay. What is your preferred approach of pre-selecting?
I don't know, we've done a change here, for example. 5-stage analysis we thought now, and
Yeah.
but here you have question marks. Still working on it.
Okay, thank you. Any other, so there's one question online, let me just catch up with this. Oops. So, someone just used, I'm sorry that I just initials for the name, so I don't know who this is, but they are saying thanks for the nice talk. And have you considered using either a full epistasis GWAS model or variance QTA method for mapping for the mapping in your simulated data?
There are epistatic software.
Yeah.
For example,
Yeah.
the method described, Luis Varone described quite a few of them.
Hmm?
We don't want to use an epistem because, as I said, it's very computer intensive. We want to find something that is more empirical.
Yeah.
And filter these interactions actually in an additive model. We don't need to change the whole setup. work what we have. As Robert says, we don't need to be exact in our description of the variance, just improve what we have. So if you go to epistatic models, they're quite heavy. Of course, afterwards, we can compare if we develop something more empirical with the epistatic models. That's the next step, to compare it, how well it works, compared with an epistatic model. But because they're already developed, we were thinking to develop something empirical.
Yeah. Thank you. And, maybe this is, maybe I missed it, but so the interactions you fitted, they were between chromosomes.
Yeah.
Why is there a reason why you didn't consider effects or within chromosomes?
We try to keep it, the first one, to keep it as plain as possible.
Okay.
So
Okay.
that's the reason. Actually, we can fit as many as we want, but see in the simple scenario what's happening, and then move to more complex scenarios.
Okay. Okay. Makes sense. Thank you. Any? Okay.
Just an idea. I work with microbiome and we have many variables and we use partial squares. It's like PCA but based on, but you can fit the latent components that best fit with a variable that can be your trait in this case.
Yes.
And the advantage versus linear regression is that not only identifies the one important but all the group that are important, but I don't know if it will support the amount of variables that you have. I think people has made GWAS.
It's one way to make selection.
Yeah, one
of your markers.
to
Yeah.
filter and then you get PLS.
Yeah. That's one option to select, to pre-select some markers, and in order to reduce the size also of your comparisons.
Yeah, I know it supports GWAS, but I don't know, but with two interactions, and you have too many variables.
Yeah. One option is to filter them first based on the additive and the pervasive, which is going hand by hand with pervasive epistasis, and then see the interactions. If you reduce the number of SNPs, you might be able to get a second threshold to select those that they might interact with each other.
Maybe.
So you can do two-stage selection, for example.
Or work with haplotype blocks and then
Yeah.
Hmm.
But there are many different options.
And you can combine several options.
Yeah. In the one or two, we think in the 5-stage analysis, one is to use haplotype blocks. Haplotype blocks.
Okay, thank you. Any other remarks or questions from? Yeah.
I'm interested in the simulation program that you have. I'm curious, when you say that it's computationally intensive, what do you mean exactly by that?
If you have, for example, 500,000 interactions from 1,000 SNPs, okay, you pre-select your SNPs that you describe your variation. That's the best SNPs that you describe most of your variation. And you want to identify, for example, which of these is additive from those SNPs and which one is epistatic. If you could try to fit interaction only in 1,000 SNPs, only the second order interactions are 500,000.
So that's what you're saying is we need a lot of time.
If you go third order interactions, even more. So it's increasing a lot. As I said, for people that have the computer power in big clusters, it's easy, but for poorer people, it's not so easy.
So one way to address that would be not to fit interaction effects, but to fit the epistatic values of the individuals, like we do with the GRM for the additive part. So we're estimating the reading values,
Yeah.
not the latest substitution effects.
Yeah.
So I think we can do the same for the epistasis, which collapses your 500,000 to
Yeah.
number of individuals.
Yeah, they call it this way. They call it. Remember the term now. Done in human genetics. That's one method. That's one method.
So you could fit it on a laptop.
Yeah.
It's not too big. Right?
It's the principle is again with building haplotype blocks, in principle, because you merge them together.
You mean that
In basic principle.
you mean that the say that epistasis effect can be large, but the variance explained is very small. And if you actually fit the variance component of epistasis, generally you might miss it because it could be a large epistasis effect, but the variance explained is so small that you don't get it sometimes. I don't know.
You don't pick it up because it's too small. You cannot. You won't pick it up. You won't pick it up in a variance component method.
That's what I actually say. That's what
Yeah.
one of the reasons that people say that why to bother much about epistasis when they explain very little the variance. Once you fit one, you realize the effect is large if the variance increases. But yes, in a variance component, that how you might know see it.
But the variance component is going to be small, but the individual interaction effects will be estimated, I would imagine. They'll be shown, but they'll be shown for you to the table.
Yeah.
Sorry, I'm just. George has made a remark that it's a bit difficult for the people at home to hear the questions, but I just wanted to say we are having more a discussion right now, so I don't really want to interrupt and repeat everything. So I'm sorry for everyone at home, but yeah, maybe we find a solution for this the next time. So far, I don't see any more questions online and nothing here from the room. So thank you again very much.
Thank you. It was a pleasure.
Yeah, it was a pleasure for us as well, and it's really, really nice to be back in